In today's webinar, Dr. Jackie Damon, Scientific Director of Stem Cell Technologies Contract Assay Services, will talk about lineage commitment by using EPO and erythropoiesis as a case study. Let me now hand you over to Dr. Damon.
Hello, welcome to Stem Cell Technologies and Contract Assays webinar, a guide to erythropoiesis, EPO signaling, and erythroid development. Today I will discuss the hormone erythropoietin and the process of erythropoiesis. More specifically, I will first cover the biology of EPO, including the regulation of erythropoiesis by EPO, and then spend a little bit of time talking about EPO binding to its receptor. Finally, I will review both in vitro and in vivo functional assays that can be used to detect the functional activity and potency of erythropoietin. To begin, erythropoiesis is part of the hematopoietic process by which stem cells divide and differentiate to produce the multiple types of mature cells found in the blood. This process is regulated by signals from small polypeptides called cytokines. Here we see in this diagram is a cartoon of hematopoiesis illustrating how a small population of relatively quiescent hematopoietic stem cells located in the bone marrow can produce the billions of mature cells found in the blood. The characteristics of these very important stem cells are that they can self-renew to maintain stem cell populations, as well as proliferate and differentiate to eventually generate all the mature cells in the blood, including the myeloid, erythroid, and lymphoid populations. A true hematopoietic stem cell is ultimately defined by its ability to in vivo repopulate the hematopoietic compartment of a compromised host. This process is regulated and controlled by small regulatory proteins called cytokines produced and secreted by a variety of cells in the body. Cytokines can act on cells within the hematopoietic system that express the appropriate receptors on their cell surface. In response to these regulatory proteins, cells expressing their receptors are stimulated to proliferate or differentiate. If we look at the action of cytokines on the various hematopoietic pathways seen in this slide, we can see that some cytokines are involved in multiple pathways. IL-3, for example, is important in the proliferation of early as well as later progenitors in different cell lineages. Other cytokines are more specifically involved in regulating the proliferation and differentiation of specific cell lineages, such as erythropoietin or EPO, seen on the left. EPO is the only cytokine absolutely required for the final stages of red blood cell development. In adult erythropoiesis, unlike embryonic erythropoiesis, which occurs in the adult bone marrow, and it is a process by which mature red blood cells called erythrocytes are derived from early progenitors called erythroblasts in the final stages of development as depicted here on this slide. This process is characterized by ribosome synthesis, the production of hemoglobin, and finally the ejection of the nucleus, resulting in a cell well suited for the job of carrying oxygen to all the cells in the body. This process is regulated by two things. First, the level of erythropoietin in the serum, and then the level of expression of the erythropoietin receptor on the target progenitor cell population. The expression of erythropoietin receptor is restricted to very specific cell populations downstream of the hematopoietic stem cell, and the level of receptor expression decreases as erythroid progenitors differentiate. The amount of EPO produced and subsequently available in the circulation is important to the regulation of erythropoiesis, and this slide depicts how EPO production is controlled. As one might guess, EPO production is actually regulated by oxygen concentration in the blood. There are regulatory DNA sequences upstream of the gene for EPO that are hypoxia response elements, or HREs, as seen here upstream of the target genes. These elements are the binding sites for hypoxia-inducible transcription factors, or HIFs. Only under hypoxic condition is HIF-1 alpha able to enter the nucleus, where it can heterodimerize with HIF-1 beta and CBP. This complex then stimulates EPO production, as well as other genes important for survival under hypoxic conditions, like genes to improve oxygen flow, including angiogenic factors, for example. Other transcription factors can also regulate EPO gene expression. GATA1 has been shown to inhibit EPO gene transcription by binding to EPO the EPO promoter under normal oxygen conditions. To recap, EPO production is increased in response to hypoxic conditions, and EPO is the only required cytokine for erythroid progenitor differentiation. When starting with the hematopoietic stem cell, EPO was involved in the proliferation as well as the differentiation in combination with other cytokines, as these other cytokines, specifically IL-3 and stem cell factor, are important factors that synergize with EPO and mediate proliferation, specifically of the earliest stem cells, until the EPO receptor is expressed on these cells. Let's now take a look at EPO, the molecule. This slide illustrates the protein structure of EPO. The majority of EPO is produced in the kidney, mostly from endothelial cells in the kidney proximal tubules. Smaller amounts of EPO are produced by other cells like liver and tissue macrophages. Erythropoietin is a 3,400 molecular weight glycoprotein consisting of 165 amino acids. Much of the complete molecule is made up of carbohydrates, as depicted here in red. EPO is in fact about 40% carbohydrate attached to the protein via 3 glycosylation sites. These carbohydrates are very important for the activity of EPO, as unglycosylated forms of EPO are quickly cleared from the body, resulting in these forms having little or no in vivo activity. EPO potency is typically measured in units. One international unit of erythropoietin has historically been defined as the amount of EPO that produces the same in vivo stimulatory effect as 5 micromoles of cobalt. It can also be measured as the amount of EPO that is required to produce the equivalent traded thymidine incorporation into erythroblasts as 1 unit of the World Health Organization EPO reference standard. EPO concentrations are typically expressed as units per mil to take into account that different forms of EPO have different specific activities. For example, the specific activity of recombinant human EPO is approximately 125,000 units per milligram for the unglycosylated protein, while urinary EPO has a specific activity of 70,000 units per milligram. I will cover how functional activity and concentration of EPO can be measured later in this talk. Recombinant human erythropoietin has been one of the most successful therapeutic agents, as it has been used clinically to treat anemia resulting from renal failure. Since EPO is made in the kidney, renal failure results in a lack of production of endogenous EPO, the consequence of which is anemia. Recombinant human EPO was first approved for human use in patients with chronic renal failure by the FDA in June 1989, and in the last 19 years has generated billions of dollars in revenue. Prior to the production of recombinant human EPO, transfusion was used to treat anemia, but in 1989, Amgen launched the first recombinant EPO that was sold under the trade names Eprix in the US and Procrit in Europe. Other clinical uses of EPO include the treatment of anemia due to HIV, autoimmune disease, myeloproliferative syndromes, and cancer. In each of these, anemia is the result of a decrease in the number of erythroid progenitors, which becomes depressed as a result of treatment with drugs that are toxic to these progenitors. More recently, EPO has entered into clinical trials as a neuroprotective agent, and there is now a growing body of evidence to show that EPO has a protective function in traumatic brain injury, as reviewed in this recent article in Developmental Neuroscience. Interestingly, EPO has been found in cerebrospinal fluid, indicating, at least in the case of injury, that EPO can cross the blood-brain barrier. Underlying mechanisms believed to be involved in EPO's protective effects include decreased susceptibility to glutamate toxicity, inflammation, and nitric oxide-mediated injury, as well as antioxidant effects and protective effects on glial cells, and of course, improvements to blood flow. Improvements to EPO to prolong its activity has continued to be the focus of many drug companies, and there are now new second-generation EPOs. Amgen, for example, has now produced darbepoetin alpha, which has a longer half-life in the circulation due to 2 additional glycosylation sites that were engineered on the EPO molecule. However, due to a decreased EPO receptor binding activity, the therapeutic advantage of this modified version has not been as dramatic as hoped. Roche, on the other hand, has modified EPO by adding polyethylene glycol groups, producing continuous erythropoiesis receptor activator, or SERA. This has dramatically prolonged the half-life of EPO in the circulation for up to 135 hours, resulting in less frequent dosing despite a similar low binding activity to the EPO receptor. Other ways people have tried to improve EPO activity are to produce synthetic EPOs like the polypeptide SEP that is similar but not identical to recombinant EPO, as was done by Griffin Therapeutics. To date, however, experimental animal data looking at the circulating half-life of this version have shown variability, so this version has not yet entered clinical trials. More recently, some non-EPO mimetics have been successful as erythropoiesis stimulating agents. The advantage of these types of molecules are that they are not immunologically related to EPO, and therefore, for patients that develop anti-EPO antibodies and subsequently red blood cell aplasia, these molecules can be used for treatment. In fact, the first one on this slide is the first mimetic antibody or mimetabody developed to mimic the effects of EPO. Mimetabody technology is new, and the idea behind the technology is to use the FC portion of an antibody fused to a protein of interest. These molecules have some of the advantages of antibodies, like a long serum half-life, as well as easy expression and purification. In particular, CNTO528 has been shown to have more prolonged reticulocytosis as well as a rise in hemoglobin following IV injections than darbepoetin. It has also been shown to be successful when administered as an aerosol. In a very recent publication in 2009 at a group in clinical pharmacokinetic at a group at Johnson & Johnson, where they evaluated this molecule in 24 healthy patients, they found that it had twofold higher activity than the standard recombinant human EPO, so studies on this molecule as a therapeutic will likely continue. Hematide is another EPO mimetic and was co-developed by Affymax and Takeda Pharmaceuticals. Phase 3 studies recently completed with hematide has found it to be effective in the treatment of patients with chronic kidney disease. Originally, hematide was identified by a peptide screen by Affymax, where they identified a short peptide not related to EPO in amino acid structure, but a peptide nonetheless that demonstrated very good EPO activity. Advantages of an EPO mimetic over EPO is that as a short peptide, hematide could have greater stability, lower immunogenicity, be simpler and cheaper to manufacture, and most importantly would not cross-react with anti-EPO antibodies, allowing treatment of patients with red cell aplasia. Other strategies that have been investigated as alternatives in the stimulation of erythropoiesis that are not based on stimulation through the EPO receptor are HIF stabilizers, which affect EPO production, and inhibition of the hematopoietic cell phosphatase downstream of the EPO receptor. With regards to HIF stabilizers, the idea is that by stabilizing hypoxia-inducing transcription factors, one promotes more EPO expression and ultimately more erythropoiesis. Although this works quite well, and in vivo testing resulted in observable increases in plasma EPO, it is uncertain what other genes would be upregulated, as HIFs are upstream of many genes. Despite this, Phase 2 trials were conducted with the compound known as FG2216, but due to one female patient who developed fatal hepatic necrosis, the future of this compound is uncertain. Another novel approach is to inhibit the off signals downstream of the EPO receptor by inhibiting the hematopoietic cell phosphatase, HCP, also known as SHP1. This protein has the opposite function to kinases that are known to turn biochemical signals on in a cell following binding to the receptor. HCP, or SHP1, is responsible for regulating signals downstream of EPO binding to the EPO receptor and turning these signals off by removing the phosphate group added by kinases. In fact, due to many years of scientific research, we know that HCP is a tyrosine phosphatase which dephosphorylates the major EPO activated kinase JAK2. The idea of using an inhibitor of HCP came from a recent publication in 2004 which showed that the EPO responsiveness of CD34 hematopoietic stem cells was affected by expression levels of this phosphatase in a dose-dependent manner. Using this inhibitor as a treatment for anemia is still theoretical, however, and testing in humans has not yet begun. I will just take a few minutes to now explain how EPO binding to its receptor actually leads to proliferation and differentiation so that you can see how and where one can design targets to influence erythropoiesis. The act of cytokine binding to a specific receptor on the cell surface initiates a biochemical process we call signal transduction. This process transmits signals from the outside of the cell to the nucleus and thus directs the cell's decisions to proliferate or differentiate. The process of signal transduction first involves activation of the receptor, which typically occurs following ligand binding by internal receptor tyrosine phosphorylation. Subsequent proteins are attracted to the phosphorylated receptors and are themselves activated. The associated proteins can then activate proteins downstream, eventually leading to modifications in transcription factors that can enter the cell nucleus and regulate expression of genes required for either proliferation or differentiation. There are several kinds of receptor families, and this slide shows some general classes of hematopoietic cytokine receptors. Class 1 and Class 2 receptors are the cytokine receptors of the hematopoietic superfamily and are characterized by some common features. An extracellular domain that is similar to fibronectin, as seen here, and which also contains distinct patterns of cysteine amino acids, as seen by the horizontal black lines. Also characteristic of Class 1 and Class 2 receptor superfamilies is a complete lack of a kinase domain in the cytoplasmic tail. The cytoplasmic region of these receptors contains instead a characteristic box 1 domain to which an intracellular associated kinase can bind. The difference between Class 1 and Class 2 receptors in this superfamily is that Class 1 contains a characteristic stretch of amino acids called the WSX-WS motif, which is important in regulating receptor dimerization when ligands bind. The important thing about cytokine receptors is that they span the cell membrane and are the mechanism by which cytokine signals are transmitted from the outside environment through the cells, resulting in downstream biochemical changes that are transmitted into the cell nucleus where decisions to divide, differentiate, or survive are made. Expression of these cytokine receptors is also very restricted in number and progenitor type, resulting in another level of regulation of the hematopoietic pathway. If we look specifically at the receptor for EPO, we find that it belongs to the Class 1 cytokine receptor superfamily, and the EPO receptor is expressed as a protein that ranges from 66 to 78 kilodaltons when analyzed by SDS-Page. And one molecule of EPO activates erythroid cells by binding to 2 EPO receptors, thereby resulting in the dimerization of the EPO receptor. EPO binding to EPO receptor changes the conformation of the receptor, resulting in activation of the associated internal kinase, the JAK2 kinase. This then leads to EPO-induced intracellular signaling, which occurs through rapid tyrosine phosphorylation of several downstream proteins. This next slide is an illustration of the EPO receptor, and when EPO binds to the receptor, it homodimerizes, leading to 2 JAK2 kinase molecules being brought in close proximity so that they can cross-phosphorylate each other. This leads to their activation and subsequent phosphorylation of the 8 tyrosines found in the C-terminal tail of the EPO receptor. Once these tyrosines have been phosphorylated, downstream signaling molecules can be recruited to the receptor through protein binding domains that bind phosphorylated tyrosines, and then the signaling cascade begins. In 1995, we set out to determine if tyrosine phosphorylation of EPO receptor was important to EPO-induced phosphorylation. We used cell lines that we constructed to be dependent on EPO for growth and infected these cells with normal or mutant constructs of the mouse EPO receptor where all the tyrosines were mutated to phenylalanine. We engineered these cells to express equivalent numbers of either wild-type or null mutant EPO receptor and asked how well these cells could grow in physiological concentrations of EPO. As you can see here, Looking simply at the cells per mil each day following addition of EPO, the wild-type cells grew well in response to 50 milliunits per mil of EPO, while cells expressing the null EPO receptor did not. On the left, we see examples of plots where we stimulated cells expressing wild-type or null EPO receptor with EPO at various concentrations to look at which downstream proteins were affected. In these phosphotyrosine western blots, we can see several phosphorylated proteins in the wild-type cells, including EPO receptor and JAK2. In the absence of tyrosines in the null mutant, phosphorylation was severely depressed, and further examination of the STAT5 protein, seen here in the STAT5 IP, one could see that following stimulation with physiological concentrations of EPO, STAT5 is not phosphorylated in the null mutant. We ultimately showed in this paper that phosphorylation of the EPO receptor is important for EPO-induced proliferation, and this tyrosine-dependent JAK-STAT pathway is still considered an important pathway in EPO receptor signal transduction and has been shown to be absolutely critical for proliferation, for example, in stress-induced erythropoiesis. This next cartoon highlights the signaling molecules involved in maintaining appropriate homeostasis in erythropoiesis by turning EPO signals off as well as on. These molecules bind to the EPO receptor to the phosphorylated tyrosines, thus putting them in close proximity. The main function of the tyrosine phosphatase SHP1 or HCP is to dephosphorylate the JAK2 kinase, thereby turning off signals downstream to the STAT proteins. The importance of normal signal transduction being initiated by binding of the growth factor to its receptor and the regulation of turning signals on and off can be highlighted in this next slide, where we see that when this goes wrong, disease is often the consequence. In erythroleukemia, for example, a missense mutation in the EPO receptor leads to EPO receptor dimerizing without EPO, resulting in constitutive activation of erythroid progenitors and a prolonged on signal. In myeloproliferative disorders, which are pre-leukemic diseases, overproduction of red blood cells is the consequence, specifically of polycythemia vera, which we now know is the result of the mutation in the JAK2 kinase. As a consequence of this mutation in the JAK2 kinase, there is constitutive activation of JAK2 and hyperphosphorylation of STAT5, resulting in hypersensitivity of erythroid progenitors to EPO, almost as if there was a perpetual on signal. This throws off the balance of erythropoiesis ultimately, and the result is too many red blood cells, which can be dangerous and the reason why historically acute PV has been treated by phlebotomy or bleeding. The discovery of JAK2 involvement in PV has recently opened the door for targeted drug therapies. Screening for these drugs has involved in vitro colony-forming cell cultures to evaluate the specificity of toxicity for these target drugs. I will now switch to discussing hematopoietic in vitro colony assays for the remainder of the talk, and will review how one can measure EPO potency and activity using these assays. According to FDA guidelines, EPO activity in vitro should be assessed in a short-term culture, specifically using erythroid tissue. The measurements can include either iron 59 incorporation into heme, incorporation of radioactivity precursors into RNA, DNA, or glycosylated side chains, or the growth of erythroid progenitor colonies from marrow erythroblasts. These are called CFUE, or colony-forming units erythroid. The FDA guidance for measuring EPO activity in vivo specifies that the bioassay should measure the biological activity of EPO in an intact animal in which endogenous erythropoietin production has been suppressed by fasting, induction of polycythemia, or exposure to atmospheric hypoxia. The effects of the test material ultimately on the rate of red blood cell production should then be directly related to the amount of erythropoietin injected, and the effect can be estimated by the reticulocyte count or the percentage of iron 59 incorporated into peripheral red blood cells. These tests are obviously labor-intensive and difficult experiments to do, so I will now take a few moments to discuss how the colony-forming assay can be used to evaluate the functional activity of EPO, both in vitro as well as in vivo. In the case of measuring EPO activity in vitro, one can do a proliferation assay using tritiated thymidine uptake into DNA. This type of assay can be very sensitive and will measure EPO-induced proliferation via binding of EPO to the EPO receptor, but it does require that the cells used express the EPO receptor preferably at a similar level to that seen in primary erythroid tissue. Two human cell lines have gained popularity for this purpose as they express EPO receptor on their surface. These are the TF1 erythroleukemic cell line and the UT7 human megakaryoblast leukemic cell line. Unfortunately, these cells are either not originally from erythroid tissue or from diseased erythroid tissue, and as a consequence may not be very representative of primary erythroblasts. In many cases, cell lines often overexpress as well as co-express many other receptors, which again often results in them not being very representative of expression of EPO receptor in primary erythroid tissue. A subclone of the UT7 cell line has been generated called UT7-EPO, which was obtained by growing UT7 cells exclusively in EPO for 6 months. The resulting UT7-EPO line has downregulated its expression of GM-CSF receptor, making it better for testing EPO, but one may still get false positive results due to expression of other receptors. A better alternative for this type of in vitro testing is to use primary mouse erythroblasts, and one can get relatively pure populations of these cells by first treating mice with phenylhydrazine for 2 days before harvesting spleens. This is also known as the Crystal assay, as it was first described by Jerry Crystal in 1983. Another way to measure EPO concentration that is both sensitive and specific for EPO is the EPO ELISA. The ELISA is most often used to measure the amount of EPO in a sample, and it is important that the amount of erythropoietin determined by this method is compared to appropriate reference standards. Analysis by ELISA is very sensitive but is dependent on the availability of the immunological active site that the EPO antibody recognizes. This has become a challenge with modified EPOs where glycosylation or pegylation may cover the immunological reactive site. ELISAs also would not work when you are looking at peptides or mimetics that are not immunologically related to EPO. Finally, one can measure EPO activity by a more functional test, the colony-forming cell assay or CFC assay. The advantage of the CFC assay is that it uses primary erythroid tissue and measures both proliferation and differentiation in response to EPO simultaneously. This assay can be used as an in vitro test or as a readout of the in vivo effects of EPO, and I'll just spend the next few minutes defining CFC assays and describing how they are done and used to measure the functional activity of EPO. This cartoon depicts the hierarchy of cells in the hematopoietic pathway again, and in this case I have inserted pictures of colonies that result when progenitors are plated in semi-solid media and allowed to proliferate and differentiate into the mature cells they are capable of becoming in optimal cytokine conditions. The key is that a hematopoietic progenitor isolated from bone marrow, peripheral blood, or cord blood has an inherent potential when plated in defined semi-solid media to divide and differentiate, forming a cluster of mature cells that can be morphologically identified under a light microscope. To perform a CFC assay, quite simply the cells from a hematopoietic cell source, cord blood, bone marrow, or peripheral blood, are prepared and at an optimal cell concentration inoculated into media that consists of a semi-solid methacellulose matrix and the appropriate cytokines. When the media is dispensed into 35 millimeter non-tissue culture treated dishes and incubated at 37 degrees and 5% CO2 with 95% humidity for 7 to 14 days, depending on the progenitor type and the species you wish to examine. After culturing, the colonies are scored based on morphological recognition of the cells within the colony by light microscopy. On this slide are pictures taken down the microscope using either the 10 or 4 times objective to give you an idea of what the colonies look like. At the top we see an example of a large erythroid colony derived from a BFUE or burst-forming unit erythroid, which you can see consists of many mature, small, reddish-colored cells. As is typical for these colonies, one cannot distinguish individual cells within the colony. Below is an example of a smaller erythroid progenitor, the CFUE or colony-forming unit erythroid, which only has limited abilities to divide and differentiate and is therefore much smaller than the BFUE and a later progenitor. Beside the BFUE, we see a myeloid colony derived from a CFU-GM or colony-forming unit granulocyte monocyte macrophage, which we can see from this picture is very different. The mature myeloid cells are clear, Their membranes are quite distinguishable, and the individual cells are larger than we see in erythroid colonies. At the bottom right, we see a colony that has both cell types. This colony is mostly erythroid and surrounded by myeloid cells on the periphery, and thus the progenitor that sat down in the semisolid media and made this colony was capable of differentiating to both erythroid and myeloid cells and is referred to as a CFU-GEMM or mixed colony. On this slide, we are looking specifically at erythroid colonies derived from mouse bone marrow. On the right, we see the smallest and most mature progenitor, the mouse CFUE, which is shown here at a very high magnification. As you can see, these are a cluster of very tiny rose-colored cells that seem to fuse and appear almost encapsulated in a shiny bag. On the left, we have a mouse BFUE that, if you look at very closely at high power, you can see consists of many similar-looking clusters of cells. The key is this is what progenitors look like in optimal cytokines. If you're interested in looking at the effects of a test compound that you think has specific cytokine activity, one can limit the cytokines you add into the media. If you look at the data on this slide, for example, and the photos of typical erythroid colonies below, you can see in cultures in optimal cytokine conditions, those which contain 3 units per mil of EPO, you get very large erythroid colonies, which are compromised in size as the EPO concentration is reduced. This size is not the only thing affected by EPO concentration, and we can see from the graph showing the total number of erythroid colonies on the y-axis and increasing concentrations of EPO on the x-axis that when we titrate into cultures increasing concentrations of EPO, we can see a nice dose response in erythroid colony number to a maximum colony number per cells plated, as well as maximum colony size. On its own, however, EPO is a poor stimulator of erythroid proliferation, and as you can see in this next figure, there is an observable dose-dependent increase of erythroid progenitor numbers in response to just EPO alone, the gray line at the bottom. But the size of the colonies are small, as indicated by the photo highlighted by the gray arrow. To evaluate cytokines and or mimetics, then, that synergize with either or both erythroid proliferation and or differentiation, one can use the CFC assay. If you now add IL-3, for example, in an EPO dose response CFC assay, you can see that you get more erythroid colonies, as indicated in the graph by the orange line. And in the photo, in the presence of optimal IL-3, as indicated by the orange arrow, the size of the colony does not really change, thus suggesting IL-3 synergizes with EPO to improve the number of progenitor colonies able to grow out, but not the number of cells per colony or the proliferative potential of the progenitor. In this next slide, you can see what happens when we add IL-6. There is no increase in either erythroid colony number or size, suggesting that IL-6 does not synergize with regards to either EPO proliferation or differentiation. In this slide, however, we are showing data in which we added saturating levels of stem cell factor, which obviously synergizes with EPO. We can see from the graph that we get more erythroid colonies, and in the photos with optimal stem cell factor, indicated by the orange arrow, we can see that the sizes of the erythroid colonies are substantially larger. We can also see from the photos that as the concentration of EPO decreases in the presence of stem cell factor, the size of the erythroid colony also decreases, suggesting that stem cell factor's main activity is to support stimulation of maximal erythroid cell proliferation as defined by the levels of EPO and the numbers of erythroid progenitors. When you add both stem cell factor and IL-3 with increasing concentrations of EPO, you can see a further increase in the number of colonies, but the size is similar to what we saw with just stem cell factor. What this shows is there is a maximum size that erythroid colonies can be in a given culture, as well as a maximum number of colonies based on the actual number of progenitors plated in the original sample. More cytokines can result in increases in colony numbers and or size until this maximum is reached. The effects on erythroid progenitors can also be examined in very early hematopoietic stem cell progenitors, or what we identify for human hematopoietic stem cells as CD34 cells. An example of the actual number of colony-forming cells observed in a starting population of CD34 is seen in this table. In this example, we have used CD34 positive cells purified from human bone marrow and added increasing concentrations of EPO in the presence of suboptimal concentrations of the synergistic cytokines IL-3 and stem cell factor. The result is a nice dose response in the total number of erythroid colonies, again in increasing concentrations of EPO as we expect. What one can also measure in this experiment with the way this assay is designed is also the ability to simultaneously measure activity on myeloid progenitors. As seen in this table, there are a number of myeloid colonies observed due to the presence of the supporting cytokines IL-3 and stem cell factor, which also support myeloid progenitor proliferation. Therefore, if a test compound is added to this media and has the ability to stimulate erythroid or myeloid progenitor proliferation, in the case of stimulating myeloid progenitor proliferation, you would expect under these conditions to also see an increase in the number of CFU GM colonies as well. In summary, this data shows that CFC assays can be used in drug development to screen for new cytokines and cytokine mimetics, as well as screen for new compounds or new and improved cytokines that have direct effects or synergistic effects both on proliferation as well as differentiation of the various hematopoietic progenitors, including specific effects on erythropoiesis. Although more difficult, it is ultimately very important to also be able to measure EPO in vivo activity. The potency of EPO in vivo can be very different than what one sees in the potency of EPO in vivo can be very different from what one actually sees in vitro due to the fact that EPO can be cleared quickly from the body. Despite the FDA requirements for using mice in which endogenous EPO levels have been reduced by hypoxic conditions, investigators have looked at in vivo effects of EPO in mice in normal oxygen conditions. Looking at iron 59 incorporation into peripheral blood is difficult to do, so other methods investigators have used are examination of reticulocytes in the peripheral blood of mice, as well as examination of hematocrit and the number of CFU-E in the spleen. Ramos et al., for example, in 2003, looked at reticulocytes, one of the tests suggested by the FDA for measuring EPO activity in vivo, but did so following EPO injection in normal mice. They showed a nice dose response increase in response to EPO and reticulocyte number in these normal mice, as seen in this dose response curve. Others have also shown that one can evaluate the in vivo effects, as I said, of EPO in normal mice by measuring the increased number of CFU-E in the spleen. Based on this, we performed our own in-house experiments in mice in normal oxygen conditions to determine the effects of injecting human recombinant EPO into mice. In this experiment, different concentrations of EPO were injected subcutaneously into groups of 5 mice for 2 consecutive days. At day 4, mice were sacrificed and the number of CFU-E colonies in the spleen, as well as reticulocytes in the peripheral blood, were analyzed. As you can see in the last column, you can see a dose-dependent increase in the percentage of reticulocytes in response to EPO, very similar to what Ramos showed in 2003. If you look, the other thing we could show was a dose-dependent increase in the number of cells per spleen, as well as the number of the more mature erythroid progenitors, the CFU-E, per 10 to the 5 cells plated, as expected in response to EPO. By calculating the total CFU-E per spleen, we could see that indeed there was a 10-fold increase in the number of erythroid colonies per spleen compared to the untreated mice. These studies show ultimately that in vivo testing for EPO potency can also be performed in normal mice. In addition, we found that EPO was responsible for mobilizing hematopoietic progenitors from bone marrow to spleen. In this next slide, we see that the BFU-E erythroid colonies, when you look at the total numbers of these per spleen, increased following treatment with EPO. But if you look below at the total BFU-E per femur, these progenitor numbers dropped. Similarly, as seen in this slide, the myeloid progenitors in the spleen also increased in response to EPO treatment, whereas the CFU-GM progenitors per femur dropped. This is not unexpected, and others have shown that EPO, like other growth factors and certain chemokines, is also known to induce hematopoietic stem cells to mobilize from bone marrow into peripheral blood and spleen. In summary, the activity of any EPO mimetic can be tested in vivo and in vitro using CFC assays as a readout. These assays have distinct advantages such that such as the fact that they do use easily accessible and appropriate primary cells and allow one to measure both proliferation and differentiation simultaneously. Another advantage is that the CFC assay tests the functional activity of mimetics, which is important when these molecules can no longer be read out in ELISAs, and these assays also allow the examination of specific effects on various cell lineages, sometimes simultaneously. Lastly, when standardized reagents and methods are used, these assays are very reproducible and robust. I would like to conclude by reminding the listeners that this webinar is hosted by the Contract Assay Services Department at Stem Cell Technologies, where we provide fee-for-service assay design and execution to evaluate hematopoietic mesenchymal as well as neural stem cells. And for more information, you can visit our website at www.contractassay.com or email me directly. Stem Cell also provides training and support, which can be found at www.stemcell.com. I would like to end by thanking Drs. Alan Eves and the President of Stem Cell Technologies, as well as Dr. Jerry Crystal, with whom I worked as an eager postdoc interested in understanding biochemical processes in development, and where I ended up instead spending a number of years working on EPO and EPO receptor biology, which was definitely a great system to have studied, especially at the time. Finally, I would like to thank the Contract Assay team who helped generate and put together some of the data I presented here today. Thank you.